everyone. This video is going to give us an overview of how to search in the NCBI database, or what was called entrees. And we're also going to go through how you can use the filters, which were previously known as the limits, and also how you can use the advanced search protocol. So what we do at the top here, you see all databases, is what we're going to look through, and I'm just going to look through for hemoglobin, which is of course a popular protein. And if we hit return, it'll give us our search results, and these are shown in the global query page. So as we've seen before, you can see all the results for many different categories. And what we'll click on is nucleotide for our example. And that will give us our hemoglobin results. And as you can see, there are a lot of them, 204,000. So what we see on the left-hand side is first we see species, and it will tell you how many species results there are. So we have animals, plants, fungi, etc. And following this, we have molecule types, and we can see that there's genomic, mRNA, and you can also customize this. And just to give you an example, you can actually customize most of these categories, but if we look at molecule types and customize, you can see that genomic DNA and mRNA is clicked or checked, and that's what we see in our molecule types results at the moment. So if we also check the box for cRNA and click on show, it will update this and put a category of cRNA up here. Now, we don't have any examples of that because there's no numbers in parentheses, but that just gives you an idea of how you can add another molecule type to your list. So going down the list here, we have source databases, we have genetic compartments, we have sequence length, release date for when the sequence is released, we have revision dates if you want to narrow that down, and we also have other search fields that we can choose from. And just to give you an idea of what these are, if you click on choose, you'll see the search field box, and if you click on the drop down menu, you can select any one of these search fields. And then to close that, you just click on the X. Now, if we want to filter in our results, say we just want to look at mRNAs, we can click on mRNA, and you notice there are 6,859 mRNA sequences. So after we click on that, this is what we see. We see just the mRNA results. Now, that filter will be active until you hit the clear button up here. So once you hit clear, then it'll go back to the original list of results. So now, for instance, if we want to filter further within the mRNAs, we can go to, say, release date. If we click on custom range, let's say we want to look at them for the past year. We put in 2016, 2017, and as a default, we start January 1st, 2016, and we go to the end of 2017. So if we click on apply, the program will filter through those results, and it narrows the results a little bit further down to 1,846. So those are two ways to filter your results, and if you want to clear those, you want to get rid of those filters, you actually have to hit clear here, and that will get rid of the release date that we added. and it'll go back up to 6,859 results. And if you want to look at all the molecular types that you've chosen, you hit clear again, and that will take us back to where we see the gene sequences and the mRNA sequences. Now, another way to search through these databases is to use this advanced link. And if we click on advanced, here you'll see the search builder. And say we want to search for hemoglobin, but we only want human hemoglobin genes or sequences. We can put in hemoglobin in all fields, and there's always a drop-down menu here. You can select any search field for any of these fields. So we'll keep hemoglobin in all fields, and we'll search for human. So if we click search, that will do a search for us, and we will come up with 8,715 human hemoglobin sequences. Now, we can also see that there are many different types of hemoglobin genes. Here there's HBB, down here there's HBG2, HbA1. So say for instance we want to go back and search for a specific gene. We can go back here and we can say gene name. So here we can put in HBB, and here we can do organism. We can look for human again, and we'll see what this gets us. It should narrow the results. And now we're down to 215 sequences. So using the filters, using the advanced search method, we can get down to a reasonable number of sequences to look at. So hopefully this is giving you an idea of how to filter results and search.